Hello, ladies and gentlemen. My name is JC, and this is JC on a bike. Specifically, this is Journal Club on a bike. This journal club this afternoon is again an undergraduate journal club for the course that's being conducted at Pitt. Today's paper is a visual cortex paper from the Cohen lab here at Pitt. And in this paper, they incidentally address a microcircuit issue, a synchrony issue, a correlation issue that was addressed in the modeling paper from the Doiron lab a couple weeks ago. I think that was paper number 8. So that's a pretty cool coincidence in the sense of, if you can recall, the Doiron paper that we covered a couple weeks ago created models which were intended to address the issue that had been observed in a number of different paradigms experimentally, that when attention is directed, when attention is active, that correlations within a local circuit actually decrease, and attention can increase the correlation across areas that are connected and working together. So what this paper is going to do is try to look at those measurements in the visual system of a monkey and try to see if attention actually does these things. Or I guess I shouldn't state it that way, because they're not addressing Doiron's paper. They're just looking at correlations locally and looking at correlations across two brain regions that are working together to discover what attention does. And so the task is, well, I find it quite difficult to understand. I had to read it a few times, but the gist of it is in figure 1. And pay attention closely to the gray squares first. The gray squares are successive presentations of the stimulus in time. The stimuli are presented for 200 milliseconds, and then there is about a 200 to 400 millisecond delay where the screen is blank. Then they present the same 3 stimuli again. And they repeat this at random, as it shows in the picture, until at a certain time the stimulus that is cued, so you can see that in the first square, they first teach the monkey to only pay attention to one of the 3 stimuli. And when that stimuli changes, he is trained to saccade, to execute a saccade from the center gaze point to the stimulus which changed direction in the cued location. And if he did that correctly, then he was given a water reward. And he was never rewarded if he moved his gaze to either of the other 2 stimuli when they changed direction. And so I think in the text somewhere they say that they got to about a 66% accuracy rate when they became good at the task. And so the idea is that somewhere here, if they make them move to those 2 places, right? There's 2 locations, and interestingly enough, you can see in the final cartoon on the far right that they can have 2 attending locations in V1 that both project to the same region in M1, the same cell in M1, because M1 cells have a much broader receptive field. So essentially what they can do is move to Q location 1 or Q location 2, but then still be looking at the firing rate of V1 neurons, whether he's paying attention to that side or not. The V1 cells will still be in receptive field. And I'm sure I'm not explaining this well enough, but I hope I'm explaining it well enough so that you have good questions for the author. Because now the trick really becomes in figure 2, understanding how they separate attend in times from attend out times. Because that's the trick, really. You want to see the firing rate in V1 change due to attention and compare that to when V1 is not attended. And it's not all that clear to me where the non-attended time is. It could be that the unattended time is when they are looking at the other of the two queued locations, where the M1 or the MT cell, sorry, I said M1, MT cell overlaps there. It's not really clear to me, but this is a central point in terms of understanding the paper, is how are the attend in and attend out times determined for these cells so that we get the effects of attention. But you can also just trust the authors, but I think it's worth peeling back those layers a little bit with the author discussion. So in figure 2 shows the attend in and attend out positions or data in summary, showing that individual firing rates increase in the attended state, but the local correlation between those firing rates decreases. And that's shown in both MT and in V1. So that's pretty cool. It's worth noting that in the example that they show for V1, there isn't a significant increase due to attention, but when they look at the population as a whole, I think that's A versus D, that the population as a whole reveals the increase in firing rate during attention. I'm not sure why we're having an attack of the black birds here, but there are an awful lot of black birds here in that tree up there too. Wow, that's weird. So I've seen it before here though. It's like a grackle festival in Pittsburgh oftentimes. I think those are grackles. But so then figure 3 will show you the cross area correlations and show you actually that the cross between areas, so between V1 and MT, the correlation increases during attendance. So that's pretty cool, and that again goes along hand in hand with Doiron's modeling paper, which suggested that this would be the case. And so interestingly enough, another thing that the Doiron paper suggested was that the projection from the higher visual region should target the inhibition in the lower brain region, right? So MT should project to interneurons in V1 if Doiron's paper wiring was correct, but we're not looking at that here, but I just thought I'd connect those two. So figure 2 shows that local firing rates increase, local correlation decreases. Figure 3 shows that inter-area correlations increase, and that's pretty awesome because that kind of shows that those two brain regions are working together and better during attention, and that's a very cool observation. She didn't even look. She just started advancing. And that guy, where are you going? So I have a little trouble with figure 4 and 5. But it seems what they're trying to do there is dig into the details of how the correlation between V1 and M1 or MT arises. And it would seem to have more to do with the receptive fields of V1 rather than the direction sensitivity of MT. That's figure 4, and then I think figure 5 is showing something about time and how, regardless of when you look near the attention change, the correlation across areas is always higher. And then I think one of the coolest figures in this paper is figure 6, because here what they do is sort of test this increase in correlation artificially by driving V1 with an electrode. And so if the mechanism by which attention works in the visual system is such that it just increases the correlative activity between, where are you going? It's red. So that's really cool, because here what they do is you use microstimulation in V1 and then look in MT. And then what they do is really cool, is that they tell the animal to pay attention in the visual field of these V1 cells, but then stimulate them as well. And show that when the animal is attending to the right place, the stimulation reaches MT better and causes a higher correlation with MT than without stim. But I think it's mostly whether the signal is seen in MT or not, and the signal from V1 is amplified in MT during attention. I think that's what this figure 6 shows. So this is a pretty cool paper. I was surprised by it. I guess I got the manuscript and I didn't think it was going to be very much, but only because I guess I'm just dumb. But I think it's a really cool paper with a really cool result, and I love the fact that it seems to sing along nicely with Doiron's paper, which I also enjoyed a lot and didn't necessarily expect this complementary in vivo experimental paper to go with it. That was running a red light. Holy cow! I mean, it's incredible. This is what you're dealing with in Pittsburgh, though. It's like a little bit of a small town, so people drive a little bit like they're in a small town. You know, you're not in a hurry, so I'm in a hurry. I can run the red light, and you won't care. It's kind of where the Pittsburgh left comes from, too, I think. It's an interesting town, that's for sure. A mix of Trump country and urban liberalism, I guess, or something. So that's the paper. I don't think I need to say much more. I got done before the 40th Street Bridge, which is pretty cool. I'll probably just end it on the bridge then, the video, and then I'll hit you up at home. And I don't think there's a whole lot to wrap up. I think it's a pretty straightforward paper. I think the main thing to grasp and the main thing to dig into with the author is to try and figure out how the attend in and how the attend out times are determined. And then, have them really explain to you how they justify this calculation, because the critical thing is to convince you that when the attention is there, the attention is there, and when the attention is not, it's not, but they're still looking at the same place. Remember? So this part of the task is still not completely clear to me how that works. But I don't know how they get the measurement of attend out. I'm looking at but I'm not attending to, or you're not looking at it, so you're not attending to it. That's what's not clear to me, and something that I think you can get out of the discussion with the author. And it's important to get that out of the discussion with the author, because these kinds of tasks, the most important thing is to understand what the task is actually doing, what the animal is actually experiencing in the task. Come on, humans, let's go! You know, wait 10 minutes for somebody to take the opening, and you are parked in the middle of the frickin' road. You got the charger, not me. I'm going right there, so don't frickin' run me over, fool. Hey, thanks for joining me on my ride. That was a paper by the Cohen lab at Pittsburgh, looking at correlative differences during attention in the visual cortex in two different regions, V1 and MT. I liked the fact that this paper kind of paralleled the Doiron paper that we discussed a couple weeks ago, and I hope I gave you enough material so that you can grill that professor a bit more about how attend in and attend out times are calculated, and how the actual when he's attending and when he's not is defined. I found it a little difficult with my cursory read for this ride, so I think the challenge is still left there for you to do it, and I'm glad I got this video done a little early. Hopefully I can get it up on Saturday so you guys have a few days to watch it, instead of like last week where you only had one day. Thanks for tuning in, and if you liked it, like and subscribe. There's a couple regular journal clubs coming soon. I've already shot the footage, but I just haven't edited it. I'm getting terrible at this. But hopefully I'll get back up to 3 videos a week. Thanks again. Bye-bye.